Mrs. Chairman, dear colleagues, it's my pleasure to be here with you today. I would like to thank Professor Dr. Hala Gabr for inviting me. Actually, my talk will be a very basic overview on mesenchymal stem cells for cardiac regeneration, and the wonderful talks we have heard this morning will leave not so much for me to talk about. As you all know, cardiovascular diseases are the leading causes of mortality, morbidity, and impaired quality of life. The pathological cardiac ischemia triggers a succession of events leading cardiomyocyte hypertrophy, which culminates in heart failure and eventually death. A typical human myocardial infarct involves the loss of 1 billion cardiomyocytes and 2 to 3 billion mostly endothelial cells, leading to a significant negative impact on the length and quality of life. Despite the development of a variety of treatment options, heart failure has failed to inhibit myocardial scar formation and replace the lost cardiomyocyte mass with new functional contractile cells. So the heart have got a limited ability for self-regeneration. That's why novel approaches have been established, like gene and cell-based therapeutic approaches. The goals of these cell-based therapies will to provide a renewable source of proliferating functional cardiomyocyte, as well as trigger the neovascularization in order to provide a novel network of blood vessels. The cell types candidate for myocardial repair, the stem, progenitor, and mature cells that can induce this mechanism in vivo, starting from the embryonic stem cells to MSCs, hematopoietic cells, bone marrow cells, and ending by the induced pluripotent stem cells are all candidate. Adult stem cells, as we all know, exhibit a limited capacity for differentiation, and the bone marrow represented the classical adult stem cell source. MSCs in the bone marrow actually are 0.001% of the nucleated cells, and they reside virtually in all postnatal organs. Friedenstein et al. In 1970 described that bone marrow contains a population of hematopoietic stem cells and an infrequent population of stromal cells that displayed adult stem cell multipotency. MSCs have got very interesting properties. They can be easily isolated from the variety of tissues and culture ex vivo. They have high expansion potential in culture, up to 50 cell doublings. They can be differentiated under appropriate in vitro culture condition to adopt cardiomyocyte and vascular cell phenotypes, and they can be delivered systemically by intravenous injections. They have also other properties like genetic stability. They can be readily transduced by a range of vectors and retain their transgene. Compatibility with tissue engineering principles, the potential to trigger regeneration in various fundamental tissues, including the myocardium, the ability to home to the damaged tissue or inflammatory sites, and they have the immunoregulatory properties as well as the immense paracrine functions, as has been said this morning. They can be differentiated into several types of tissues, and this is a tubular cartilaginous construct engineered from amniotic cell MSCs. Sources of MSCs, adult bone marrow, this is the first one, derived MSCs. Then we have the adipose tissue derived MSCs, umbilical cord derived MSCs, and the fetal tissue derived MSCs, Wharton jelly, amniotic membrane, etc. The identification and characterization of MSCs depend on the International Society for Cellular Therapy, plastic adherence to cell culture dishes, specific surface antigen expression patterns, that's to be negative for the hematopoietic CD45 and CD34, but positive for the MSCs CD15, CD90, and CD73 markers, as well as differentiation into mesoderm, chondrogenic, adipogenic, osteogenic lineages. These are the culture, primary culture, and explant cultures and confluent cultures. So this is the high expression level of the adhesion molecules as well. MSC's immunomodulatory properties, they, as have been said, they stimulate Tregs, suppressor, some cytokines, suppress escape T cell recognition, and they express low levels of the major histocompatibility MHC class 1 molecule and do not express MHC class 2 or the co-stimulatory ligands CD40, CD40L, 80, or 86. So they have been recognized for their possible role in treatment of graft versus host disease. Now, to promote cardiac tissue regeneration, we need sufficient numbers of cells to be delivered and maintained within the heart at the site of the left ventricular dysfunction. And the new tissue needs to be vascularized electrically and mechanically coupled with the rest of the myocardium. In the functional and structural effect with the reverse remodeling, scar size reduction, increased tissue perfusion, improved regional contractility, and increased ejection fraction. Homing of MSCs depends on the presence of cytokines, chemokines, growth factors, and interactions with integrins and selectins. When cells remain in the heart, which cells end up in other organs as a result of a washout effect? Stem cells injected locally within the heart are also observed in the lungs, spleen, liver, and kidneys after a few hours. One day after transplantation of neonatal cardiac myocytes into the rat, only 24% of the originally injected cells remained in the heart. PET scanning and MRI have been tested recently in humans to track stem cells. 1 hour after injection of the fluorodeoxyglucose labeled CD34 cells, only 5.5% of cells were detectable in the heart by PET scanning. This is labeling, tracking, and differentiation of stem cells. Experimental animal studies have been investigated since long time. The swine model was one of the very important one and proved to be safe, effective in improving left ventricular function and increasing myocardial tissue perfusion. This picture shows MSC's reduced perfusion defect in the ischemic pig heart and enhanced collaterals in the infarcted area. Myocardial delivery of cells, there should be sufficient initial retention of the cells and adequate local engraftment. So that's to transplant enough cells into the myocardium at the site of injury to maximize restoration. There are several methods: the intracoronary infusion or catheter-based intramyocardial needle injection, or now, this is the most important, the direct intramyocardial injection during surgery. Autologous bone marrow-based cell therapies for cardiac repair has been used extensively. They were safe in the randomized controlled trials. I will not name the clinical trials, but usually the data support a small yet significant increase in left ventricular ejection fraction in cell-treated patients. This is isolation of stem cells from the bone marrow. The difference between autologous and allogeneic bone marrow-based cell therapies for cardiac repair: the allogeneic, of course, this is from young donors, so they have a higher regenerative potential, and they need no bone marrow collection. While the autologous, they are rare among the bone marrow and need to be expanded ex vivo, and the procedure takes days to weeks. The progenitor cells from old patients have reduced proliferation potential, and they need GMP cell practice. Following the clinical trials, many other clinical trials have been used, especially the Poseidon and Prometheus studies, allogenic and autologous bone marrow, or autologous bone marrow for patients undergoing bypass surgery. Most of them showed significant enhancement in clinical performance and ejection fraction. However, the results are all inconsistent. But MSC therapy is safe and devoid of adverse reactions like arrhythmias and so forth. Off-the-shelf allogenic MSCs like the Osiris and Mesoblast product have proven safe in phase 1 and 2 trials in patients with acute MI.